Good evening, everyone. Welcome back to our Neurotorium educational series. And this week we are looking at Alzheimer's disease and other dementias. Basically, this is the final week of our series. Yesterday, we looked at the history, the definitions, and the diagnosis of Alzheimer's disease. Basically, Dr. Muyanja took us through the definition of what Alzheimer's disease is and helped us differentiate it between uh, Alzheimer's disease and dementia in general. And he told us how Alzheimer's disease is not basically synonymous with dementia, though very many people actually tend to confuse the two or to use the terms interchangeably. But Alzheimer's disease is a distinct part of just one of the causes or the etiologies of the dementias. He took us through the symptom and clinical presentation of individuals with Alzheimer's disease, specifically the cognitive domains, deficits in the cognitive domain, where we looked at memory loss, basic forgetfulness, including even misplacing items and attributing it to probably people stealing from them, and also even forgetting, reaching a point where the person even forgets their own relatives or even themselves. Then, of course, he talked about the functional impairment that follows. This is basically the inability to perform the activities of daily living. And then he mentioned about the neuropsychiatric symptoms like the mood disturbance, the agitation, the aggression, as well as wandering. He also took us through the clinical diagnosis, all the way from what you need to pick out in the history, on the physical examination, the laboratory investigations, and he mentioned that today, if you want to diagnose Alzheimer's disease, you have to base on the biomarkers, as well as he told us about neuroimaging. Then he took us through the role of the neuropsychiatric screening instruments that we can use, the mini mental status exam to assess for cognition, as well as the Montreal Cognitive Assessment, the MOCA, can be used. Then he also took us through the differential diagnosis, possible things that could explain the symptomatology of the individuals who might present as though they have Alzheimer's disease. Today, Dr. Muyanja is back yet again to take us through the neurobiology of Alzheimer's disease. Basically, he wants to make us understand the brain mechanisms behind the symptoms that we talked about yesterday, what exactly goes on and where to lead to these pathology or these symptoms. Allow me to invite Dr. Muyanja to take us through today's presentation. Dr. Muyanja, you're welcome.
Thank you so much, Dr. Katama. Kindly confirm that I am audible enough and that my voice is not distant.
Yes, please. Yes, please. You're very clear.
Thank you very much for that recap of what happened yesterday. Certainly, it is going to make my life easy. Good evening, colleagues, ladies and gentlemen, seniors. Dr. Orikiriza Judy, I'm so happy to see you in the chat and the rest of the colleagues. Thank you for joining us in today's session where we are going to delve into the neurobiology and etiology of Alzheimer's disease. So, friends, please strap in and enjoy this journey. So, to understand Alzheimer's disease, we start with two key proteins in the brain. That is amyloid precursor protein and tau. I'm going to be using the word APP to refer to amyloid precursor protein. So, let me start with APP. APP is a normal and useful protein that is found on the surface of neurons, and it helps with neuronal growth, synapse formation, cell signaling, and it even protects the brain when it is under stress. So in a healthy brain, typically, when certain proteins age, after a certain period of time, they are supposed to get broken down and reabsorbed to be used as nutrients for formation of other proteins within the body or the brain in this case. So in the brain, we have enzymes that break APP into smaller fragments. And this is mainly amyloid beta 40 and amyloid beta 42. These ones are usually cleared out without the brain having any problem. So we have two broadly categorized enzyme systems that are very important within the context of Alzheimer's disease. I'll refer to the first one as the healthy pathway and the second one as the harmful pathway. So the healthy pathway uses alpha secretase enzymes, which are going to break down amyloid precursor protein into harmless and soluble amyloid beta 40. On the flip side, when talking about the harmful pathway, we are having beta and gamma secretases. These are enzymes. And these beta and gamma secretases together are going to cleave APP to form amyloid beta proteins that are long, poorly soluble, and tend to accumulate in the context of Alzheimer's disease. And the most common victim here is amyloid beta 42. So in the context of Alzheimer's disease, the activity of the harmful pathway or the activities of the beta and gamma secretases far exceeds the activity of the alpha secretases. And this implies that there will be an increased production of longer and less soluble fragments from the breakdown of APP, the most common of which is amyloid beta 42. So amyloid beta 42 is longer, is less soluble, and is harder for the brain to clear. So over time, as this amyloid beta 42 is accumulating in the brain, it is going to misfold and clamp outside the neurons to form what I'll be calling amyloid plaques. So when I talk about plaques, kindly know that it is from amyloid beta 42, which is folded and clamped. The second protein worth talking about is tau. So tau is a structural. If anyone is wondering, let me quickly get the pointer. Let me see. Yeah. So we've been talking about amyloid beta plaques. Now we are coming to tau. This is the spelling. So tau is a structural protein that is found inside the neuron, and it stabilizes microtubules. So when tau becomes hyperphosphorylated, it loses the function of stabilizing microtubules, and it will collapse into twisted fibers, which are given a name which keeps messing me up: neurofibrillary tangles, or NFTs, or call them tau tangles. So in the context of Alzheimer's, we are seeing two major issues. Outside the neurons, excess or poorly cleared amyloid beta 42, okay? Then inside the cell, we are seeing hyperphosphorylated tau, which is forming tau tangles. And together, these two are going to disrupt communication. They are going to damage synapses, and they are going to eventually kill neurons, leading to the slow cognitive decline that we end up seeing clearly. But what is striking is that both plaques and tangles start forming many years before the symptoms actually appear. So, while amyloid and tau are central in the neuropathogenesis of Alzheimer's disease, they are not the only explanations that we have. Along the way, I am going to explain further what happens. Neurodegeneration simply means the slow and progressive breakdown of nerve cells. And once this process starts, it cannot reverse. And over time, it ends up leading to the death of neurons. In Alzheimer's disease, abnormal proteins which I've talked about, such as the amyloid plaques outside the neurons, and then the tau tangles inside it, end up triggering a toxic cascade. So these deposits are going to disturb the normal function of the cell, are going to lead to neuroinflammation, mitochondrial problems, and metabolic stress over time. So altogether, these changes will damage communication between neurons and eventually lead them to die. We identify this degeneration in living patients through FDG PET. PET is positron emission tomography. We can also identify this through MRI, which shows brain atrophy. However, even CSF markers of tau and neurofilament light chain can actually give us an idea of what is going on in the brain. Now, let us delve into the risk factors associated with Alzheimer's disease. Alzheimer's disease doesn't come from one single cause, ladies and gentlemen. In most people, it is sporadic, and this means that it develops from a mix of many factors which are working together over time. Age, that we can see here, is the strongest risk factor, but genes and environmental influences also play a role. Now talk about head injury. Think about infections. This also plays a role. Think about cardiovascular disease. Yesterday, I talked about microangiopathic injuries as an important explanation as to why Alzheimer's may at times occur in some people. So a very small percentage of these people, that is about 1%, is Alzheimer's of a familial origin. This is where a genetic mutation is driving the disease. So from this, we can see that Alzheimer's is really the result of a number of risk factors which are converging together rather than a single trigger. Talking of genetics, genetics play a big part in Alzheimer's, although they are certainly not the whole story. We know that the disease is quite heritable, and somewhere between 58 and 79% can account for what? For this actually. A very small group, as I earlier said, have what we refer to as the familial type. And this familial type of Alzheimer's is caused by mutations in APP, presenilin 1 and presenilin 2 genes. So this occurs usually before the age of 65 years. But for most people, the gene that we pay most attention to is the APOE gene. So carrying APOE4, epsilon 4, has been found to actually increase one's risk of developing Alzheimer's. On the other interesting flip side, having APOE2 as a gene has actually been found to decrease the risk of Alzheimer's in a number of studies. But it is very important to note that research has also picked up many other risk genes, especially those that are linked to inflammation and vascular problems. So I can summarize this slide with just this sentence. Genetics loads the gun, but environment and lifestyle are the ones that pull the trigger for someone to eventually develop Alzheimer's. So when we think about dementia risk, there are factors which we can change, but there are factors which we cannot change. You will not change your genetics, however much you want, okay? So the Lancet Commission reminds us that about 40% of dementia risk worldwide actually comes from modifiable factors, 40%. And these include the things we see every day in our clinics. Low education, people think education is just for the sake of making money. They don't know that it is very important in improving your cognitive reserve. Hypertension, hearing impairment, smoking, obesity, depression, physical inactivity. All these factors we are seeing here are modifiable, ladies and gentlemen. Air pollution, head injury. There are times we think that telling people to put on helmets while riding on border borders is just for the sake. Ladies and gentlemen, head injuries have been implicated a lot in the causation as well of early onset dementias. On the non-modifiable side, age, as I earlier said, remains the strongest risk factor. And more women than men have been found to actually develop Alzheimer's. Certainly, family history matters a lot, especially in those with dominant genetic mutations. And as I said, people who carry 2 copies of APOE4, if your father and mother donate to you copies of APOE4, you are likely to show earlier abnormalities. So the risk is coming from both modifiable risk factors and non-modifiable risk factors. Alzheimer's has a very long silent phase. So what we do early in our lives and midlife actually matters a lot. Many lifestyle factors can reduce the buildup of brain pathology. Talk of regular exercise. These are things we take for granted. Good cardiovascular health, avoiding smoking, avoiding heavy alcohol use. We are moving towards salary week. Controlling hypertension, managing your weight, okay? Protecting your hearing, okay? And keeping stress levels down. All of these help to protect the brain, and certainly factors such as reducing air pollution. It is tragic that in the Ugandan context, we have not come to appreciate some of these issues that we think are benign. We have a lot of noise pollution in our society, and we have a lot of air pollution in our society, and we think, sadly enough, that it has not all on us. On the other side, there are other activities such as engaging in cognitive activities, maintaining an education, ladies and gentlemen, staying socially active, and looking after mental health. All of these will strengthen our cognitive reserve. When we are looking at dementia risk across someone's lifetime, from the time you are born up to when you go to meet your creator, you realize that it is not one single event that pushes the brain into trouble. The risks, right from the time you are born, quietly pile up over time. In early life, poor education is going to limit your brain's cognitive reserve. Friends, when I talk about cognitive reserve, I mean the brain's ability to tolerate damage and still function as fairly normally as possible. Now, as we enter middle age, things such as hypertension, obesity, diabetes, smoking, unhealthy habits, to name but a few, eventually start getting added to the picture. And certainly here also, the risk of head injury, middle age, adult life is increasing. Later in life, that is especially after the age of 65, depression starts setting in. Social isolation comes in, physical inactivity, air pollution, that is a constant in the Ugandan context. All of these increase vulnerability. And all these factors interact with our genetic background, including APOE, to shape whether someone eventually will develop dementia or not. On the other side, protective habits such as being physically active, okay, remaining socially uh, engaged, being educated, having a good diet and nutrition, okay, all of these help the brain to resist damage for a longer period of time. So I hope that someone here has understood the importance of exercise in the context of dementia, the importance of a good diet in the context of not just dementia, but Alzheimer's dementia in particular. Talking of the etiology, Alzheimer's disease has a complex etiology. Just from what I have shared, I believe you understand this. But 3 pathological processes have been found to sit at the center of our understanding of Alzheimer's. First is the amyloid hypothesis, where abnormal breakdown of amyloid precursor protein produces amyloid beta fragments that accumulate, excuse me. Yes, so the amyloid hypothesis, I was saying, is where abnormal breakdown precursor protein produces amyloid beta fragments that that, as we earlier saw, accumulate outside neurons as plaques. But second is what we refer to as the tau hypothesis, where tau proteins now inside the neurons become hyperphosphorylated, form tangles, and block the cell's transport system. So these two processes, the amyloid hypothesis and tau hypothesis, work together to drive the third feature, which is progressive neuronal and synaptic loss, especially in the hippocampal areas and the cerebral cortex. And these changes explain the brain shrinkage that we see on PET and MRI as well. And you know, the enlarged ventricles that are typical of these neurodegenerative diseases. So let us talk about the amyloid hypothesis. Today is a day for neurobiology and etiology. So there is no way we can dodge this. Friends, the amyloid hypothesis explains Alzheimer's as a problem of imbalance. The brain, as I earlier explained, produces amyloid beta. But in the context of Alzheimer's, there will be failure to clear it clearly. So it will slowly build up into plaques. And these plaques can set off harmful changes like tau pathology and neuronal injury. And this can occur long before the symptoms have appeared. So we see strong evidence from families with amyloid, with APP and presenilin mutations, where altered amyloid processing actually leads to early plaque formation. So in short, amyloid buildup is one of the earliest biological steps that pushes the brain towards Alzheimer's disease. But the other hypothesis, besides the amyloid hypothesis, is called the amyloid cascade hypothesis. The amyloid cascade hypothesis builds on the idea that amyloid beta, most notably amyloid beta 42, is the earliest trigger of Alzheimer's disease. So whether the problem is genetic, as we can see here, like APP or presenilin mutations, or reduced clearance with age, amyloid beta 42 eventually accumulates. So the small amyloid beta oligomers will interfere with synaptic efficacy over time, long before the symptoms appear. And over time, these oligomers will deposit as diffuse plaques in the cells, and they are going to activate not only microglia, but also astrocytes. Now we know that microglia and astrocytes are going to play a role in inflammation, okay? But the story is not going to end there. We are also going to have a challenge with disturbance of ionic balance along the way. And because of this, we are going to have a set of a chronic inflammatory response within the brain. And we know that with chronic inflammation comes cellular destruction. So this inflammatory environment then is going to promote abnormal tau phosphorylation, formation of tau tangles, and widespread neuronal and synaptic loss. And the end result is cognitive decline, which we diagnose as dementia. The story has not ended there. We also have what we refer to as the tau hypothesis. So I already said that tau is a microtubule-associated protein whose job is mainly to stabilize the internal transport system within the axon. In the context of Alzheimer's disease, tau is going to become abnormally or becomes abnormally hyperphosphorylated. And this hyperphosphorylation makes it to detach from microtubules. It ends up misfolding and aggregating eventually into neurofibrillary tangles or tau tangles. And once these tangles form in vulnerable brain regions such as the medial temporal lobe, the neuron loses structural stability, transport breaks down, and the cell progressively degenerate. Now, it is important to note that although some theories place tau as the starting point of disease, most modern evidence actually is showing that tau pathology spreads after amyloid beta changes have begun. It is also important for us to note that clinically, the burden of tau tangles actually correlates a lot with the symptom severity. It has been found to correlate more with symptom severity than with the concentration of amyloid plaques. And this is very important in therapeutic for therapeutic purposes. Talk about tau pathology. Tau pathology in Alzheimer's disease has been found to actually follow a predictable route. And this pattern is what we referred to, or what I referred to yesterday as the 6 Braak stages. So when tau pathology is happening, it typically begins quietly in the mediotemporal lobes, okay, where it is going to mess a lot with memory-related area, the entorhinal cortices and the hippocampus and so on. From there, that is why actually one of the first symptoms within the context of Alzheimer's most times is memory issues. So from here, the tau pathology is then going to spread into the temporal and association cortices. And finally, it is going to reach the neocortex in the later stages. So it spreads from within outwards. This tau spread has been found to correlate much more closely actually with how a patient declines than the amyloid explains. So to understand this kind of progression that we are talking about here, the Braak stages, we rely on 2 major types of tau biomarkers. That is the CSFP tau shows us abnormal tau that has leaked out of damaged cells. So it directly reflects the underlying disease process. Tau PET shows us where the pathology has accumulated over time. It shows you how much damage has already been done. So together, CSF and PET give us a fuller picture. CSF is telling us the biology that is happening now, but positron emission tomography, PET, is showing that tau has left in the brain. In Alzheimer's disease, we now see that the interaction between amyloid beta and glial cells is actually central to neuroinflammation. So when beta amyloid first appears, we saw earlier that microglia actually activate, as we can see here. And the activation normally is to protect the brain. But as beta amyloid continues to accumulate, these same activated microglia and astrocytes, by the way, become less effective, and they are now going to shift into a state of chronic inflammation. Studies show that when amyloid beta deposits, tau and activated microglia cluster in the same region, tau actually tends to spread much faster. So the meaning behind this is that beta amyloid buildup and microglial activation start separately, but they eventually converge to drive the neocortical tau pathology. And a lot of lab studies have actually been found to support this. In Alzheimer's disease, the brain slowly develops a kind of energy failure, if I may say that. So normally, insulin and like growth factor, IGF-1, help the brain cells to grow, to repair themselves, and they play a critical role in communication between cells. But in Alzheimer's disease, it has been found that the brain actually becomes resistant to these almost the same way the body becomes insulin resistant in the context of diabetes. And when that happens, the brain starts using energy poorly. If we are conversant with diabetes, I think we understand this well. So over time, brain metabolism is going to slow down. And this happens way long before anything can be seen on MRIs. And that is why studies such as FDG-PET may actually be very important in the diagnosis of Alzheimer's, especially if it is early onset. Now, this chronic insulin resistance is going to not just stop there, it is going to fuel a dangerous cycle. We are going to have more inflammation, we are going to have more oxidative stress, the mitochondria are going to have a lot of struggles, and we know that once mitochondria are affected in any process, you end up with oxidative stress, and certainly we are going to have challenges with handling of lipids. And because these changes usually appear after amyloid and tau have already built up, many scientists now actually describe Alzheimer's as partly a metabolic disease of the brain. So we need to talk about development and importance of biomarkers for us to understand the neurobiology of Alzheimer's disease. So I have talked a lot about biomarkers, but when we talk about biomarkers in Alzheimer's disease, we are simply talking about biological signals that tell us what is happening in the brain before we can even see it clinically. So a biomarker is any objectively measured indicator of a normal process or an abnormal process, a disease process, or the body's response to treatment. For Alzheimer's, these markers come in two main forms. We are going to have neuroimaging, such as MRI or positron emission tomography. But we are also going to have fluid markers from CSF or the blood. For Alzheimer's, these markers come from imaging, like MRI or PET, and from fluids such as CSF or blood. Fluid biomarkers can actually tell us whether someone truly has Alzheimer-type pathology. But imaging is going to add on to make the picture more complete by helping us to understand how advanced the disease is within the brain, and therefore we shall be able to stage. So because mild cognitive impairment can come from many different causes, it is very important, ladies and gentlemen, to know that biomarkers can help a lot to reduce misdiagnosis and even to do away with unnecessary investigations. So this brings us to the question yesterday, which a colleague asked us about whether we can go ahead and do investigations without biomarkers. The truth is yes. Currently in Uganda, we are doing we are managing Alzheimer's and other dementias without using biomarkers. It is mainly research-oriented organizations such as IBI, to name but a few, that have the abilities or the resources to conduct research using these biomarkers at the moment. So currently, we are managing patients mostly clinically, and maybe the only biomarkers we are using are maybe MRI related, where we can see the different changes, the enlargement of the ventricles, the cortical thinning, and so on and so forth. So currently, within our context, unfortunately, we are limited, but we are studying not only to work in Uganda, but all over the world. And so we need to have this knowledge. Now, when we are talking about biomarkers in Alzheimer's disease, we now use a simple ATN framework. Let us follow this together, okay? A stands for amyloid, which I have talked about exhaustively. Amyloid is measured through CSF or positron emission tomography. The T stands for tau, which is also assessed through CSF or tau PET. The N represents neurodegeneration, which we can pick through MRI atrophy, FDG, PET hypometabolism, or total tau in the CSF. So this ATN framework helps us to describe what is happening biologically in the brain long before symptoms appear. So the more widespread the pathology on these markers, the more advanced the disease stage is. But we need to remember that this biological staging is mainly a research tool at the moment, unfortunately. Some people with elevated amyloid or tau may stay cognitively normal for years. This is an interesting finding. So whenever we have such findings, if you're working in the US or the UK, wherever you will be, always interpret these results in context and in relation to the history and clinical examination you would have done. The graph we are seeing here is showing how different Alzheimer's disease biomarkers become abnormal at different times as the disease progresses. On the y-axis, we are seeing the biological change over time. And on the x-axis, we are seeing how the disease evolves, the clinical journey from the silent preclinical stage to mild cognitive impairment and to full-blown dementia. So the first measurable change, usually, what we are seeing here is usually a drop in CSF amyloid beta 42. And this has been found to happen many years before symptoms. And this is because amyloid beta is actively being deposited in the brain, and so less will be in the CSF. But the story is not ending there. Some of it actually is being actively pulled out of the CSF and being deposited around the neurons. So after this drop has happened, that is in CSF amyloid beta 42, next, amyloid PET is also going to become positive, okay? So after deposition, the amyloid PET will become positive, showing visible amyloid plaques. Next, after amyloid changes have happened often, tau-related abnormalities are triggered off. So we note that CSF tau actually increases, indicating early tau pathology, okay? And showing, and then later on, tau PET also becomes positive, okay? So that shows the spread of tau tangles in the brain. Now, as tau spreads in the brain, markers of neuronal injury and synaptic dysfunction are going to eventually appear on the FDEG PET, okay, over time. So whenever there are such appearance on the FDG PET, it shows reduced, the FDG PET, ladies and gentlemen, allow me a second to take a drink of water. Okay. All right, thank you. So I was talking about the FDG PET. The FDG PET shows reduced glucose metabolism in areas of the brain that are affected. But after changes have been registered on this FDG-PET, next we are going to end up seeing changes on the MRI, that is brain atrophy. But this is usually a late change. And after that, cognitive symptoms and functional decline will appear. But that is only after these biological changes have built up over years. So this graph is demonstrating that Alzheimer's has a long hidden phase, and each biomarker becomes abnormal in a sequence which is actually predictable. As we are drawing to a close, we now understand that Alzheimer's starts quietly. It can even start 15 to 20 years before symptoms show. We have also realized that biomarkers help us to detect these changes early enough, long before someone starts getting memory problems. And this matters. That is because once the brain has crossed a certain neuropathological threshold, treatment may no longer actually be able to reverse the damage that has been done to the brain. But the story doesn't end there. Biomarkers also help us to stage the disease more accurately so that we know when a treatment is likely to work best. And finally, having Alzheimer's disease specific biomarkers actually supports more personalized care. And because they guide the development of precise therapies that are tailored to the biological stage of the illness. Friends, in summary, Alzheimer's disease, we have noted, emerges from a mix of interacting factors and not just a single cause. We have also appreciated the fact that genetics play a role, and APOE4 allele remains the strongest known genetic risk factor in older adults. We also know that amyloid buildup starts early, way long before symptoms. And although tau is more closely tied to the clinical picture of how a patient presents, both processes, that is amyloid buildup and tau pathology, play or shape how the disease unfolds eventually. We have also appreciated the reality that biomarkers have transformed what we know about Alzheimer's completely. They help us to identify the right patients for disease-modifying therapies, which I talked about yesterday and which I will exhaustively talk about tomorrow, the likes of aducanumab and so on. But it doesn't end there. These biomarkers also guide clinical trials for people who are interested in research, and they allow specialists to diagnose Alzheimer's disease more accurately in vivo. So gone are the days when we had to wait for someone to die and we do an autopsy and we do a post-mortem rather before confirming whether the cause of death was truly Alzheimer's or not. We can now make in vivo diagnoses. They also help us to spot people in preclinical stages. So in simple terms, as we are walking away from this discussion, biomarkers have strongly shifted Alzheimer's disease from guesswork to clarity. And if we understand the disease earlier, we can actually fight it better and earlier and allow people to live with dignity. And ladies and gentlemen, that is where the hope lies. Thank you so much for listening to me. Dr. Katama, back to you.
Wow, wow, wow. This has been epic. I always enjoy these sessions because of the much information that we actually get. I thank you, Dr. Muyanja, very much for making this session quite interesting, simple, and easy to follow. You always make things easy to understand, no matter how complicated they might seem, because when you're reading about this neuropathology of Alzheimer's disease, of the dementias, you can get lost in those proteins, the tau, the amyloid, beta proteins, and others. But you have made this quite easy to follow, and I have personally enjoyed, and yes, my knowledge has again increased. This has been our session 2 of the 3 for this week, the final week of our Neurotorium educational series. Tomorrow, same time, same link, we shall be looking at the management principles. Please join us. We shall not be just looking at the management, but the management principles. We want to understand what has to be done when managing individuals with Alzheimer's disease, and what some guidelines say about the management of Alzheimer's disease. And maybe Dr. Muyanja will give us insights on what we are doing in our setting. Do not forget, our sessions are always shared on, we upload them on YouTube, so we shall be sharing the link to that. And also more of these presentations. Of course, for us, we add just these presentations to fit our program, but more of these and more literature is actually available on the YAM website, which is free, accessible. You can go there, you sign up for the newsletter so that you can always get updates of whatever is going on in the world of science of these conditions. I hope, Dr. Muyanja, you have taken a minute or two to sip a cup of water. So we are here with questions. I see a couple of them, the chat, and I'll be reading out one so that I give you time to respond. Thank you, Luisa Fernandez, for joining us from Portugal, and everyone else who is with us today. Joy Naiga talked about the PhD, that she's coming for it. Whether she wants to study Alzheimer's disease in particular or not, I don't know, but probably that's why she posted it here. Please, you're welcome to the world of science. Sarah Nassos asks, which genes are most commonly mutated in familial Alzheimer's disease cases? Dr. Muyanja.
Thank you so much, Nasozi, for that question. I earlier actually talked about it, that there are mainly three. Mainly is the word. The first is APP, which directly affects how amyloid precursor protein is processed. But we also have presenilin 1 and presenilin 2. Okay, so the presenilin genes, which are part of the enzyme complex, how should I put this? The presenilin genes are important in determining how the enzyme complex that will cut up APP develops over time. So when someone has any of these three mutations, APP, presenilin 1, presenilin 2, the brain is going to end up overproducing the toxic amyloid fragments that I talked about. They having any of these genes is going to cause an overdrive of the beta and gamma secretase enzyme systems. Briefly, that is what I can say.
All right, thank you for that response. Then, yes, Dr. Kemirebe greeted everyone, those from Portugal, from Congo, and even those from Sunday Apple asks, is there a link between phone emissions and neurodegenerative diseases? Dr. Muyanja, do you have something to say about this?
Hey, so comrade, we have read a lot of literature, and to be honest, there are no studies yet that have shown a clear increase in Alzheimer's, Parkinson's, or other neurodegenerative diseases because of phone use. Yes, it's true that some animal and laboratory studies suggest biological effects of radio frequency radiation, but the doses and conditions often do not really match real-life use. So the findings are inconsistent. So I cannot speak authoritatively about that. There is no clear literature linking the two. Thank you.
Right. Thank you very much for that response yet again. Yes, Dr. Musa Mahmoud is saying that thank you very much. He's thanking the entire Uganda Neurotorium Organizing Committee and, of course, the presenter, Dr. Muyanja, for today's insightful session. And he says that as we approach the second last day of this training program, we would like to respectfully raise the following oriented questions regarding the neurobiological mechanisms underlying Alzheimer's disease. Like we said, these are educational series. That's why you always find quite interesting questions that need to be answered because, yes, people are being educated and we need to understand this information. The first question says, Dr. Mian, could you elaborate on how the dysregulation of the amyloid precursor protein processing leads to the accumulation of the neurotoxic amyloid beta 42 peptides, and why this shift from normal to pathogenic cleavage remains a central driver in Alzheimer's disease pathophysiology? Dr. Muyanja, I guess that you first respond to the first question and then we shall go to the second question.
Thank you so much, Dr. Katama. Dr. Muse, we appreciate these insightful and thought-provoking questions as usual. You have been a valuable attendee of these sessions. Thank you so much. So for this question, the problem begins when the normal processing of amyloid precursor protein shifts towards the amyloidogenic pathway. Instead of APP being cut into harmless fragments, APP will instead be cleaved by beta and gamma secretases into long and mostly insoluble fragments that are going to aggregate over time around cells. That is not the end of the story. These fragments that are produced are actually stickier and have been found to be more prone to forming oligomers that over time disturb synaptic communication. With time, these toxic clusters or the plaques end up triggering inflammation. As I explained earlier, within my explanation, I talked about them activating microglia and astrocytes, which initially are supposed to help with dealing with any form of inflammation, but over time, they become less sensitive and descend into a state of chronic inflammation. And certainly we know that in the context of inflammation, we are having a lot of oxidative stress, an environment, in summary, which is toxic to the survival of the cell and leads eventually to neuronal degeneration and death. Basically, that is what I can talk about. So the challenge is with the shift towards the amyloidogenic pathway. Instead of producing particles from the breakdown of APP which are soluble, because of overactivation of beta and gamma secretases, we end up having production of particles which are long, insoluble, and which eventually accumulate in around cells, wreaking the havoc that I have talked about.
Yes. Thank you, Dr. Muyanja. So the second question from Mohammed Musa was, considering the role of neuroinflammation, how do the activated microglia and astrocytes contribute to both the progression and the potential mitigation of Alzheimer's disease, and what therapeutic implications this does dual role present? Yes, Dr.
I think the second question actually builds on what I was already saying. Okay. The reality is that microglia and astrocytes in the brain sit at the heart of inflammation. So initially, when you are having deposition of beta amyloid and so on, and they are activated. They help by clearing amyloid and, helping with tissue repairs and maintenance of homeostasis. But as this disease progresses is going on, they will eventually, as I stated, become chronically activated. They will release cytokines and oxidative molecules that are going to worsen the already existing damage. So this double nature is why actually therapeutic strategies need to focus on gently modulating rather than suppressing of glial activity. At the end of the day, the aim is to preserve the protective role of these cells while still limiting their, how can I state this, while still limiting their harmful of activation that could eventually fuel neurodegeneration. That is the best way I can respond to Dr. Musa's question. Dr. Musa, I would like to meet you in person. I would like to shake your hand for those wonderful questions. It has been quite wonderful. Thank you.
Yes, thank you, Dr. Muyanja, and thank you everyone who has tuned in. I think I'm going to invite Dr. Mwesiga to just have a word or two towards the end of this today's session. Dr. Manuel Mwesiga.
Okay. Thank you again. Sorry, it took a bit longer to unmute, but I think I want to start by also thanking Dr. Muse. First of all, I would like to congratulate you. I saw somewhere on social media that you are graduating with a Master's of Medicine in Psychiatry, and I think based on the questions, I acknowledge your teachers who did a good job. So I want to congratulate you. I would also like to congratulate everyone. Tomorrow is the last day. It's been 7 weeks of a lot of knowledge. We hope we can continue this going forward, but let's meet tomorrow and finalize with treatment and hopefully show you a bit of the data that we have gathered over the last 7 weeks. Thank you.
Yes, thank you, our PI, for leading us through this entire training, all the way from the beginning to this point. Everyone is really thankful of Dr. Muyanja's delivery the information. Brian Mulinda, you're saying that you request the presenters to drop the slides. So these slides can be found on YouTube, but also the presentations that we've made can be found on. However, when you go to neurotorium.org, let me type that link. So when you go to neurotorium.org, you're going to actually find under the themes, you're going to find the different themes of all these conditions. So here we have looked at mainly those that are within the spectrum of psychiatry, but you're going to find even those that are within neurology. You're going to find presentations about things like migraines and others. So you're going to find all this information on the Neurotorium website, which is actually free of charge. So you can just go there, you can download, you can use them as long as you recognize that these materials were obtained from the Neurotorium website. Thank you everyone who has tuned in today, and I would like to invite you tomorrow to the closure of our series for this year. It has been a wonderful ride. We've had indeed a lot of knowledge that we've learned, and I invite you all to tomorrow's session. Same link, same time. See you tomorrow. Have a wonderful evening.